everyone. Thank you for joining us for the Cannabis Testing and How to Read Test Results webinar, presented by NCIA's Scientific Advisory Committee. My name is Alena Rodriguez. I'm the Managing Director at ARM3 Labs. I started off as a microbiologist and testing samples and doing method development. And then I led our process to become ISO 17025 accredited. And now I'm managing daily operations for our lab in Boulder, Colorado. I'm joined here today with two other members of the Scientific Advisory Committee, Tiffany Coleman, who's the Director of Quality at Copperstate Farms, a medical marijuana agribusiness in Arizona. She brings 15 years of pharmaceutical experience to the cannabis industry and has expertise in good manufacturing practice compliance, quality assurance and quality control, project management, and research and development. Also with me is Garrett Cropsey. He's a project manager at Canna Advisors, where he provides support for client business ventures in cannabis and hemp across the US and abroad. His background's in ecology, pharmaceutical manufacturing, and cultivation and dispensary operations. And his expertise includes testing requirement, quality assurance and quality control, standard operating procedures, and operational efficiency. So without further ado, we're gonna jump right in. So on our agenda today, we have two topics we're going to cover. The first topic is discussing what tests we're performing and why. And then we're going to launch into how to interpret test results and provide some examples of test results. So I'm going to pass over our discussion to Tiffany to start us off with what tests we're performing and why.
So we're going to start with why are we testing? This is a pretty simple thing across the board. We're testing because people matter and the people using our products have a right to know what's in them. What are those people? Those people are medical patients. Those people are recreational consumers. It's also anyone else that might be in the process, any handlers across the board. If somebody's touching our product, they have a right to know what's in it. The foundation of good manufacturing practices is really based on CISPQ. CISPQ means safety, identity, strength, purity, and quality. The first four actually hold up quality the entire time, making sure that the product is always the same, that we know that what we're producing is not going to harm anyone and is safe. That's the first part. This is really highly controlled by the government, the FDA and the USDA. In our case, this is done by the municipalities and by the state legislatures. It's a little bit less. This also comes into play for efficacy. We all know that there's plenty of clinical trials out there right now trying to prove just how good cannabis is compared to other drugs that are available on the market. The next part is identity. Identity holds up quality by saying, hey, what we're saying is in this package is what it is. And this comes back to not just that, hey, this is cannabis, right? But it also says this is cannabis, it's a distillate. This is cannabis, it's a distillate, and it's an edible, and that edible has these three other components, and that's what these are. It's specific to what's going on the label and what you might have heard as label claim. Beyond that, we have strength. Strength is incredibly important in our industry because we really care about potency. It's important in lots of other places as well. So when we talk about strength, we're looking at the potency analysis, potentially the terpene analysis, and we're also making sure that what we're putting on the label in regards to strength is actually what's present. Many places in the legislature are defining this, saying that you have to be spot on, otherwise you're in a situation where your identity is being compromised and you're mislabeling. The next one is purity. Purity means free from all contaminants, right? So it is what we say it is 100% of the time. That's important for our consumers because a lot of these contaminants are things that an immune compromised medical patient is not going to want to have in their system, but it's also not good for any of our veterinary patients or any of our recreational consumers either. This could be things like heavy metals, pesticides, and even normal things like hair particles and dust from the ceiling or rust from old pipes. All of these things come together to hold up quality, and quality really again being that we are knowing what is in our package every single time, and our consumer can know without a shadow of a doubt that what they're getting is consistent across the board based on what's identified on the label. There are many types of testing. Each of these types of testing really go back to show that we're upholding our CISPQ. we're going to talk about all of these a little bit longer. So we have our composition testing, which is what is in the flower itself, the cannabinoids, and then we have our two different kinds of contaminant testing, microbial and chemical. So going back to potency. Potency is what we're defining our strengths. We have multiple kinds that we're showing here. Right here, the example we're providing is for THC and THCA. You can see that there are multiple methods that you can use to calculate this. Each method gets a little bit closer to the most accurate amount as far as the consumer and their experience with the total amount in the plant. The first one being no additional calculations, just adding the two together. The next one, a calculation to show that THCA does lose some weight when it's converted into THC. And the last one, that, hey, not only are you losing some weight when you're converting from THCA into THC, but sometimes you're not even doing a full conversion from those two things. This is really important because we put this information on our label in most jurisdictions. And if you're putting it on your label and you don't actually know that it's correct and you're not doing it consistently, then you're looking towards being mislabeled and providing inappropriate information to the consumer. Something else that consumers are really looking towards, because they're starting to be more interested in the entourage effect, is terpenoid profiling. Now, this is important because forever and a day, people have been looking at cannabis and deciding, I like that one based off the way it smells. And it's okay for you to follow your nose, because oftentimes, if you found one that you like that smells a certain way, if you find another one that smells the same way, it's going to give you a similar effect. At least that's the theory that we're trying to prove with all these clinical trials. We can actually identify these terpenoids by using either a GCMS or an LCMS so that we can get a solid separation. This graphic just shows you how many different terpenoids that are out there. And quite honestly, there are many others, and there are lots of times when they break down as well, depending on the temperature. Moisture content and water activity are quite important. As you can see from the water activity test, there's a number of things that happen when you have too much water activity or too much moisture content. Moisture content being the amount of water that is present, which is different from water activity because it is the amount of water that is present that will also react with microorganisms that may be present. Both of these things give us a good idea of how long our product can stick around without degrading or getting moldy. And if you see here, the closer you get to 1 in the water activity, the more likely it is that you're going to have a microbial contamination bloom. And it's also can be noted that the more moisture content you have, you can see there is actually a sweet point in there where you're preventing the yeast and mold growth, but you're also being able to maintain the plant quality at the same time. There are many methods for testing this. You can do a loss on drying test. You can use a capacitance tester, which is usually a handheld device. You can use a Carl Fisher test in the lab, or you can actually have a specific water activity analyzer in your lab to do this. Quite honestly, if you're using this and you're trying to make sure that your product is the best that it can be, doing both tests for one for moisture content and one for water activity is important, because that's going to make sure that you're reducing microbial contamination growth while also making sure that your test results for your potency are accurate. Since the more moisture content you have in your plant, the less potent it may appear because the weight is greater. And I'll turn it over to Alena, who's going to talk to us about microbial contaminants.
Thank you, Tiffany. So the first suite of contaminants we're going to talk about are the microbial contaminants, which include objectionable organisms that can cause harm when the contaminated product is used as intended. Examples of this include molds such as Fusarium, Aspergillus, and Penicillium, and bacteria such as Shiga toxin-producing E. coli, which we call STEC. It also includes toxins left over from microorganisms, such as mycotoxins. So below is just a list providing different objectionable organisms or toxins that might be present in different cannabis products. For instance, inhalables, we're very concerned with yeast and molds versus edibles, and topicals, where we may be more concerned with bacteria. So the word fungus includes both yeasts and molds. The differentiation is that yeasts are one-celled organisms, while molds grow into multicellular filaments called hyphae. You can see that in this picture. Some yeast and molds can be beneficial and have a positive symbiotic relationship with the organism they're living on, but other times they can cause serious infection and harm. One serious fungal infection seen in immunocompromised people is aspergillosis, which is caused when you inhale Aspergillus mold spores into your lungs. And the spores can infiltrate your lungs, causing damage to the lung cells. There's about a half a dozen published case studies of immunocompromised individuals developing this condition from moldy cannabis specifically. One of those cases, I'm showing 2 figures in the bottom left from a case published in 2008, where a 65-year-old man presented symptoms of aspergillosis, including fever, cough, respiratory problems. So his doctors ran a bunch of tests. One of them was scanning his chest, and as you can see in the bottom left, the yellow arrow is showing a lesion. So they took some tissue from that lesion, put it under a microscope, and what they saw was cell death, inflammation, and translucent fungal hyphae, which were indicative of Aspergillus species. And in January 2018, we had the first case of a healthy individual developing a fungal infection from contaminated cannabis, and this was reported in California. The individual developed cryptococcal meningitis, which is an infection caused by a yeast, Cryptococcus neoformans. Cryptococcal meningitis is a serious infection of the brain and spinal column. And both these cases raise concern about yeast and mold contamination in cannabis, even for immunocompetent people. Next, we're going to talk about bacteria, which are tiny single-celled organisms found almost everywhere, on all surfaces, all over your skin, inside your gut. Most of these bacteria aren't pathogenic in the sense that they don't cause disease, but there are a lot of pathogenic bacteria that we're concerned with in cannabis and cannabis-derived products. First being Shiga toxin-producing E. coli and also Salmonella. These both cause foodborne illness. And then more states are starting to expand their bacterial screening, including screenings for Staphylococcus aureus, which is a type of bacteria that actually 1/3 of people carry in their noses. And most of the time it doesn't cause harm, but other times it can cause staph infections. And also Pseudomonas aeruginosa, which can cause respiratory tract issues for immunocompromised patients. Also, this bacteria has high resistance to antibiotics, making it a serious concern. Lastly, we're going to talk about mycotoxins, which are secondary metabolites naturally produced by molds. They're not themselves alive, but they're the harmful poisons left behind by molds. This table lists some allowable levels of aflatoxins and ochratoxin A in food from different countries, as well as the limits in cannabis. Aflatoxins are known contaminants of peanuts and corn. They're produced by Aspergillus molds and known to be carcinogens. Ochratoxin A is toxic to the kidneys. It's produced by Aspergillus and Penicillium species. It's also known to be a carcinogen, which makes it a concern to our industry. So I'm going to toss it back over to Tiffany to talk about some chemical contaminants.
So if you're at all involved in creating concentrates or edibles and you're using any process for extraction that uses a solvent, you may already be aware of this. Residual solvents are what makes things, extracts, smell not quite like cannabis. These things can make our employees and our consumers sick. It's important to control solvents so that they're not harming the people working with them just as much as it is so that they're not harming our consumers. What kind of harm can we be talking about here? We could be talking about a lot of harm. And they could just make you want to throw up, but they could also make you really dizzy, cause unconsciousness, make you have a seizure, or even if you're exposed just in minute quantities, give you an increased risk of cancer. The greatest thing about this is that for residual solvents, there is well-established testing for all of the organic solvents and their degradants that can be performed using Headspace, GCMS. It is a test that's been performed for decades, and it works just as well on cannabis and cannabis concentrates and edibles as it does on all of the other things that are made in our economy. So there's 2 parts here. One, you can use it to make sure that your employers are okay and that your process is working correctly, because if your residual solvents are where they're supposed to be, then you know that your process for removing those is working correctly. Anything higher than that, it shows that you don't have the correct controls in your production environment and you're potentially putting your consumers and your employees at risk. The next part you guys probably have heard of, which is called a couple of different recalls in the last 6 months or so, is pesticides. It's worth noting that each state has its own list of things that are inappropriate to be used, but that the federal government for food products, right, so for edibles, has a broad one for pesticide as well, saying, hey, you can't use these at all, or they can't be present at all in foods. So that exists as well. So if you don't have one locally, because your state is giving you one, it is possible that you will also have one just from the federal government if you're making edibles. Potentially also concentrates because they're inhalants. And it's important to note that if you have a little bit of pesticide or fertilizer or fungicide on your flower, that edibles and concentrates are inherently more risky, because as you concentrate the cannabinoid, you are also concentrating any contaminant that's on the cannabinoid and giving that on either to your employees while they're doing the production or to your consumers, and you don't want to do that. Pesticide testing is also well established. You can do this with a GCMS-MS or an LCMS-MS. You get solid separation with either method. There are readily available standards for you to do this testing. And you should know that not doing it not only could lead to a recall like the ones that we have listed here, but it can also just cause pretty not insignificant damage to your consumers, both for their central nervous system, meaning they're going to lose motor function and potentially ability to respirate and die. Wait, they wouldn't. We don't have any that has happened, but it could happen. But we also know that the damage over time to the people who are working with these materials is not small. You know, there's a reason that we control pesticides in our country, and that's because they're pretty toxic, because they're used to, you know, make pests no longer live. The next type of contaminant we're talking about is heavy metals. Heavy metals need to be controlled because in general, they accumulate in our body. This accumulation then can get to a level where now we're actually having a symptom from the accumulation in our body. And those symptoms are pretty bad. They can lead to things like cancer, Parkinson's, Alzheimer's. In children, you can see, you know, decreased levels of learning ability, problems with understanding and short-term memory. So these are some pretty big things, and we want to make sure that we're not causing an issue with our product, because sometimes the people who are taking these are children and other immune-compromised individuals who are already struggling with their own symptoms. The next part for our specific industry is that cannabis itself is a hyperaccumulator, which means if you're using and growing cannabis in an environment where the water or the soil that you've chosen has heavy metals, that it's going to actually take those up into the roots and the stalks, and then it will be present in larger quantities than might otherwise be the case because of the specific type of the plant. Don't have to feel crazy about this. Heavy metals testing has existed, again, for a long time. There are well-defined, standardized methods for testing this, including doing ICP-MS, which is the international standard for heavy metals testing at this point. There are many other methods where you can do benchtop digestion, atomic absorption. These are a little older, they're not as precise, but at least you'll be able to know that, hey, you know what? I don't have any lead in my cannabis. So the key points of what I've just tried to cover is that we're testing because people and animals matter. Right? Our consumers are the reason we're doing what we're doing. You know, we also do it for our employees, but it's really the people who are taking our medicine that we want to keep safe. We know that we're keeping them safe by using CISPQ. CISPQ means that we are creating a holistic plan around quality to support it. There are many different ways for us to support CISPQ with testing, including calculating potency, calculating terpene quantities so that we can be certain of our clinical entourage effects and efficacies, testing for chemical and microbiological contaminants, both for shelf life stability to say, hey, you know what, the process I used was good enough to remove all the microcontamination, and that stuff's going to be good for 6 to 9 months or a year or 3 years. While also making sure that we're not creating additional symptoms for our consumers, whether they're a recreational consumer or a medical consumer, from the contaminants that we're putting in our product. That's definitely something that we don't want to do. And by doing appropriate quality controls on the front end and doing product testing on the back end, we can meet those two things in the middle and prevent any of those negative outcomes for our consumers.
So now we're going to lead into our second half of our presentation on how to interpret test results. So I'm going to give Garrett this.
Awesome. Thanks everybody for joining us today. The first portion that we'd like to discuss as part of the second half of our presentation is the difference between quantitative and qualitative measurement. So quantitative measurement is regarding the quantity of the analyte in question. It produces a numerical value for that analyte quantity. Therefore, it can be measured, and it's asking, is it there? And if it is, how much is there? An example of quantitative testing would be potency testing for cannabis, where they're advertising a specific percentage of dry weight for the potency of the particular flower product, or in the case of edibles and concentrates, the milligrams of THC or active cannabinoids. The other form of measurement is qualitative measurement, wherein you are assigning a quality to that measurement. It seeks to be descriptive. It's more observation-based as opposed to measurement-based, and is asking, is it there or not there? And then how do we quantify that? So, for example, bacterial testing in Colorado is merely a presence-absence or a pass-fail. So one of the ways in which quantitative measurements inform qualitative measurements is that you can use a quantitative result to describe a qualitative measurement. So for example, if you observe anywhere between 5 and 10 units of an analyte, that could be equated to a medium density of observed quantity. Therefore, a result of 7, the quantitative measurement, would be reported as medium density, a qualitative measurement. So before we get too much further into this section, I'd like to go over a couple common terms that we'll be using. I've already stated analyte, which is what is being identified and measured. There's a couple different forms of units or ways of quantifying concentration of an analyte within a substance. For example, milligrams per milliliter, milligrams per gram, colony forming units per gram, which is a measurement for bacteria, micrograms per gram or parts per million, and then also nanograms per gram or parts per billion. Regarding those two in particular, just so you can get a sense for exactly the amount of analyte that we're looking for in the samples being analyzed, 1 part per million is approximately 1 1/4 2-liter bottles in an Olympic-sized swimming pool. And 1 part per billion is half a teaspoon in that same Olympic-sized swimming pool. So very, very small concentrations of these pesticides, residual solvents, et cetera. And we have equipment, like Tiffany mentioned earlier in the presentation, that can test that. So related to the abilities of that equipment, as well as the methodologies used to perform different tests, 2 terms that we'd use in that context would be LOD, the limit of detection, and LOQ, the limit of quantification, which we'll go over in this next slide. So regarding these different reporting limits, these reporting limits have to do with the equipment that you're testing on, as well as the methods used to test for different analytes within that sample. So anything below the limit of detection could be equated to NIGHT, which is to say that analyte was non-detectable at that level, and it is highly unlikely that it is found within that sample. Any testing result that comes in above the limit of detection but below the limit of quantification, you could equate to twilight, or it is detected, but it's so little is detected that we don't know quite how much is there, but we do know that it is there. And then finally, anything above the limit of quantification could be equated to daytime, in which we know it is detected and it is of an amount that we can be certain of the quantity. So it is there and we know how much is there as well. On this slide, we'll talk about different acceptable limits for these test results. And this varies state by state and also by intended use. So on the left-hand side of this slide, we have acceptable limits for different pesticides in different states. We have California, Oregon, Washington, and Colorado listed. And then on the right-hand side, we have different limitations set in different states for total yeast and mold, fungal species, bacterial species, total enterobacteria, and mycotoxins. So on the left-hand side, you'll notice that, for example, imidacloprid, one of the pesticides along the left-hand side, the acceptable limit in California is 5,000 parts per billion, and in Colorado, only 20. So you can see that these acceptable limits vary greatly state to state, as well as pesticide to pesticide within those states. As far as the mycological and bacteriological contaminants found in cannabis and cannabis products, the testing for those also varies greatly depending upon the location that you're working within. So you can see that in the case of, let's just take one of these, the fungal species, right? In California, they're testing for aspergillus, several forms of aspergillus, and in Colorado, they're not testing for those at all. Same with Oregon and Washington. So this is one example of what's safe and acceptable varies based upon your location, and that's one way in which the cannabis market in the US makes operating and testing for these things a little difficult or complicated, certainly, as it's up to each individual lab to understand that their equipment is capable of detecting everything that that state in particular is looking for, and within the limits of detection and quantification necessary to make sure that those tests are in fact correct. So with that being said, it's important to note that under the limit does not necessarily equal safe for this reason. These limits are a reflection of regulations and not necessarily based on safety standards, although they do all uphold safety standards. There isn't consistency within the US when it comes to what these limits are. For example, a product might pass microbial testing with a low colony forming unit count, and then given the right conditions, this relates to moisture content and water activity as we were talking about earlier, it could then fail that same test after a month on the shelf, where the microbes that were present on the sample have had time to grow to a limit that is above that initial passing result. Conversely, over the limit does not necessarily mean guaranteed harm. So, for example, if the state's limit for a substance is 0, then a result of less than 5 parts per billion would technically be over that limit. It would not necessarily mean that the product is guaranteed to be harmful. And with that, I'll turn it back over to Alena.
Thanks, Garrett. So before we get into some examples of test results, I wanted to quickly go over 3 main reasons for lab result variability, because I get a lot of questions about why labs have different results. And the first one is the sample itself. So there's a lack of standard sampling protocols in our industry. There's no statistically significant sampling model specific to cannabis and cannabis-derived products. Also, the samples themselves tend to be heterogeneous in nature. For example, flower. From bud to bud, you're going to see a different cannabinoid content, especially from the top of the plant to the bottom of the plant. And even when you try to grind these buds together to make a homogeneous sample, it's difficult because trichromes, plant material, and stems will naturally want to separate. Also, contamination is not always uniformly distributed in a harvest or production batch. So if you send 2 samples for contaminant testing to 2 different labs, you may have very moldy buds that you chose for one lab, whereas the other sample may not have as much mold in the particular buds that were selected for that sample. Secondly is a lack of standard test methods. So there's no standardized test methods specifically for cannabis outside of water activity, which was just published by ASTM. So labs are using different storage, preparation, and extraction processes For example, some labs will dry flower samples prior to potency analysis, and others will not. And the reason why some labs do that is to give a more level playing field for the samples, as there's no standard protocols for drying or curing buds. So they all come in with different moisture contents. Also, we're generally, across labs, we're generally using the same instrumentation. For instance, HPLC is the gold standard for potency testing, but different analytical parameters may be applied. We may be using different internal standards. We may have different flow rates on our columns, that sort of thing. And the last reason for lab variability is measurement uncertainty. Measurement uncertainty is the quantification of doubt that exists with every measurement, the plus or minus, if you will. So you might think when you look at your watch or you look at the thermometer in your car, you might think that it's giving you a precisely accurate answer every time, but there's always going to be a plus or minus associated with all measurements. And even if the same test method were being used across labs, inter-lab results would be similar, but not necessarily the same, because each lab is going to have a different measurement uncertainty. The goal of any lab is to decrease this uncertainty. So jumping into potency results, I just wanted to bring up again why we use this method for determining total THC or total CBD content. And that's this equation at the top. So total THC equals THC plus THCA times 0.877. And again, the reason we're using 0.877 here is to account for the molecular weight difference of the acidic form of THC versus the active form of THC. So on the left, you see THCA structure. On the right is THC. The only difference is the carbonyl group that's circled in red. And so THC weighs 87.7% of what THCA weighs, and that's why we use this equation. So this is an example of a potency result in flower. At the top of the test result, you might see the total THC or the total CBD content, along with the equation they use to calculate that. And then you might see a table. So on the very left, we're listing all the analytes. In this case, it's cannabinoids. Next to that, we're listing the limit of quantification in milligrams per gram. So that's the lowest concentration that this lab can detect for each of these cannabinoids. So if they detect a cannabinoid less than 0.1 milligram per gram, but they still detect it, you'll see a result like what you see for CBDA, under lower limit of quantification or under the limit of quantification. So it's there, but the lab can't accurately give you a result. Lastly, we see the levels of the cannabinoids presented in two different forms, milligrams per gram and then percent by weight. The last thing I'll point out on this slide is the THC conversion at the bottom. Some labs will report the THC conversion percentage, and that just means the amount of THC that was already naturally converted into the active THC form. In this case, most of the THC present is in the acidic form still. Next is an example of a concentrate result. And I chose to show a distillate result where the total cannabinoid content, as you can see at the bottom right, is over 100%. And you might think, how can you get over 100%? Theoretically, you can't, but this is where measurement uncertainty comes into play. So there is a measurement uncertainty associated with each result for each cannabinoid. And then when you add all those uncertainties together, that's how you can get an over 100% result. So again, we're seeing the list of analytes, the limit of quantifications, and the amount of cannabinoid present. And at the bottom, we see the THC conversion of this example is 100% because distillates have been treated with heat, thus converting all the acidic THC to the active THC form. The last potency example is of an infused product, in this case, a CBD edible. So the target potency of this edible was 10 servings of 10 milligrams each, totaling 100 milligrams of CBD. On the left, you see a graph like we've seen before, listing all the cannabinoids. The only difference with this result is the right graph, which is showing you the homogeneity test. So the homogeneity test is just performing the potency test on multiple servings. And in the lab, we call those replicates. So you can see here, we have 4 servings that we tested. We weighed them out and we determined what the milligrams of THC and CBD were in each piece. And then the way we determine if it passes homogeneity is to look at the relative standard deviation, which is the measurement of how similar each individual result is to each other. So in this case, this passes homogeneity testing. Also, you might see a within permitted variance pass or fail. And so in Colorado, the permitted variance is 15% for infused products. What that means is if we get a sample in the lab that's labeled 100 milligrams CBD and we test it, the result must be between 85 and 115 milligrams for it to pass. So next we're going to go into moisture content water activity results. This is where we first start to see acceptable limits. So again, moisture content, the amount of moisture in a sample, and water activity is the amount of free water that's able to react. So the acceptable limits here, for instance, in Washington is 15% moisture content and 0.65 AW units for water activity. So AW units, 0 means pure dry versus 1 means pure water. So in this case, the results passed for both, and this sample had 8% moisture content and a AW of 0.56. Below, I'm showing you an example of a moisture sorption isotherm. Tiffany showed you one of these earlier. This one's specific to cannabis samples. It was created to show the correlation between water activity and percent moisture. You can see that the upper acceptable limit of water activity at 0.65 generally correlates with the upper acceptable limit of about 12 to 15% moisture content. Next, we're going to look at a microbial result example from Colorado. So you'll see the analyte listed. So in this case, it's organisms. You'll see the acceptable limit. In this case, we're looking at colony forming units per gram of sample, and then the limit of quantification along with the result. So this is an example, unfortunately, of a failed total yeast and mold result. And most of the tests we've discussed so far have been quantitative. This is the first example with the Salmonella and S-Tech results here that you see are qualitative. So what we were doing was testing for absence or presence. And in this case, we did not detect either of those bacteria. The table below is just an interesting table that I found. Colorado State publishes every 6 months the percentage of packages that pass testing and tell us what they fail for. So if you look at this, the infused products and the concentrates are almost always passing microbial testing. Where we start to see fails is in the flour. And most of those fails, from my experience, have been total yeast and mold. Next, we're looking at residual solvents results. So again, you see a table like we saw before with potency, except we have acceptable limits here. So this is an example of the California list with their acceptable limits. And one thing I'd like to point out is I think it's very important for you to, if you're getting testing done, to consider what the limit of quantification is versus the acceptable limit. You want to make sure that your lab can detect far lower than the acceptable limit, which will help you do trend analysis as well. So this example is showing that we detected acetone in the sample, but it was under 300 micrograms per gram, which was the LOQ for acetone. So we couldn't give an exact number. And the sample unfortunately failed for butane. So next is an example of pesticide results, which is very similar to the solvent results, except the analytes in this case is pesticides. And just for sake of simplicity, I'm showing a Colorado example because we look for 13 pesticides in Colorado versus other states that might be looking more for like 65 pesticides. Again, you have the limit of quantification and acceptable limit, and you want to make sure that the lab can accurately detect down to levels that are far below the acceptable limit. And in this case, we only detected a little bit of myclobutanol, but it was under the limit of quantification. And lastly is an example of heavy metal results from Nevada. So I've chose the acceptable limits for Nevada here for the big 4 metals: arsenic, cadmium, lead, and mercury. And so this result looks much similar to the other chemical contaminants we were looking at, pesticides and residual solvents, where you'll see the analyte, the acceptable limit. Actually, this example doesn't show you a limit of quantification, which you might see. You might see that the reports don't have that. Ask your lab. They should be able to give you that information. So I'm going to pass it back to Garrett to let you know how you can use your data.
Right. So in all of these states, they require cannabis testing of all these different types be performed prior to that product being sold at a dispensary. So this gives every cannabis business on the cultivation and processing side a valuable stream of information that you can use to ultimately learn more about the products that you're making or the cannabis that you're cultivating. So establishing testing data history is invaluable to your business for several reasons. These trends can provide insight into cultivation and production methods and can be used both proactively and reactively. In the proactive case, you would use your data history to to quantify the effects of process changes on product quality. For example, potency testing would help you determine if alterations to your nutrient regimen are in fact enhancing potency of your cannabis flower. Changing little things in the nitrogen, potassium, phosphate concentrations can have dramatic effects on cannabinoid and terpene concentrations in your final product, and testing results will give you a way to look into that. The other example would be reactive use. So you can use data history to pinpoint inefficiencies or to troubleshoot failed test results. For example, if your moisture content results for cannabis flower went out of spec shortly after you hired a new harvest manager to begin work, it would be within your best interests to investigate that harvest manager's drying curing process to figure out why these moisture content results are now failing. So with that, we'll summarize the second half of the presentation. As far as key points for this and how to read test results, it's really important to know what analytes you're looking for each test. What is the lab looking for? What is your state looking for? And secondary to that is understanding the method reporting limits, the limit of detection, the limit of quantification for these different tests to be performed by the lab in your state in particular. Knowing what the acceptable limits are and what constitutes a failed result is very important to your business. On the fourth point, knowing what a detected analyte means and how to use those results. For example, under the acceptable limit does not necessarily mean safe, and there's a chance that the contaminant will increase in the future. And then finally, know how to use your test results, and that data history you can track data history over time and derive insights from those trends. And that's something that all cannabis businesses should be doing in order to better understand their cultivation methods and their processing methods.
Sweet. So we would like to thank you for joining us today. And I see we have some questions. So we're going to pull those up and start asking them.
We'll start at the top here. We just got our chat window open.
Okay, so I'm going to ask the question, let my counterparts answer, and if I have anything to add, I will. So can the heavy toxic metals enter the body through smoking cannabis?
Yes. There are actually really excellent studies about this, that whether you're using a filter, not using a filter, etc., that these heavy toxic metals, lead, cadmium, arsenic, do get transferred via smoke, which means if you're smoking your cannabis, and potentially if you're vaporizing, although the studies aren't finished for that, you can be ingesting these if they are present in your flower. That's why we don't want them there.
What typically happens when the cannabis flower exceeds the limit of pesticide, microbial contamination, and or heavy toxic metals?
So with that, laboratories are usually in very close contact with the state oversight boards, or in the case of Colorado, it's the Marijuana Enforcement Division. When it comes to failed test results, all of this is tracked within the seed-to-sale software, and the state would receive a notification when a batch fails testing, and then would follow up with the producer to investigate.
So the next question is, what variability are you noticing from lab to lab? And I'll take this one. And they asked percentage ? So as far as variability between labs, if you're thinking percentages, you're probably thinking potency results. And yes, we do see variability in potency results. A lot of that stems, like I said, from the sample itself. So the heterogeneous nature of flowers, how the cannabinoid content can change from bud to bud. So if you're giving a bag of buds to lab A and a bag of buds to lab B, they might not have the same cannabinoid content, even if you pulled them from the same plant. Also, the testing procedures being used. So for instance, keeping with the potency example, if one lab dries the samples for a few minutes before they do the analysis and another lab doesn't, the lab that does dry, they'll get slightly higher percentages because they're losing some of that extra water weight. And since we're giving percentages by weight, if you have less water, then that's going to increase the cannabinoid content. So next question is, for the homogeneity testing, you are testing in replicates. Does this mean testing in duplicate or triplicate or separate sample preps? So for homogeneity testing, each lab chooses their method of how many replicates they include. We include 4 replicates. And what that is just performing the test on separate servings. So you're doing the sample prep from the beginning on separate servings. Next question, extremely high levels of calcium and magnesium with no limits listed in a report I just received, presence of lead no presence of lead at all. I'm concerned about this, but the company just blew it off. Wouldn't this be a concern?
I can take that one.
Okay.
So calcium and magnesium aren't.
Oh, just kidding. I think they meant cadmium and mercury. Sorry.
Oh, okay. So yeah, sorry, I took those to be calcium and magnesium, but if they're cadmium and mercury, then yeah, I mean, if they have high levels and they're beyond the appropriate level, which is, I believe, in parts per billion, then yeah, you should be concerned about that because those are heavy metals that shouldn't be in place and you probably failed testing. It's possible that you're in a place that doesn't have heavy metals testing as part of its regulation, and then it's an internal quality decision whether or not you want to enforce heavy metals requirements on your own company. From a consumer standpoint and need to know, I would consider it a concern because we know that heavy metals are bad and that the 2 primary methods of consuming cannabis, whether you're smoking it or eating it, can definitely lead to heavy metal buildup in the body.
Thank you. So the next question is asking us to discuss the availability of cannabinoid and terpene reference standards, their consistency or inconsistency between vendors of these standards, and the impact this has on variability of reported results. This is absolutely true. Standards do have impact on variability. Do either one of you want to take that?
Tiffany, you might know more than I do.
Yeah, so there's 2 parts here. So it is possible that you're going to have difficulty getting availability, that a vendor might not want to sell you a standard, or that the total amount of standards that we have available in the United States is limited. In my experience, and I'm sure Alena can talk about this as well, the variability of the standards that I'm receiving is less than the variability of the instrumentation I'm using them to test on, which means it's not contributing that much to a variability in reported results. I think that's probably why she didn't include this in her test on why there are test result variability across labs. If you want to talk a little bit more about that, Alena.
I would just say that the regulators have noticed that there's obviously going to be variability, also a limited supply and vendors that are producing these standards. So that most states actually will require labs to use 2 sets of standards from 2 different vendors so that you can double check the vendors themselves. If that makes sense.
And you should always be doing supplier controls in general. That's a good quality system, which is part of good manufacturing and good laboratory practices. So if you have a vendor and you get 5 lots for them and the 5 lots aren't similar, that's an issue, and you should have a limit for that. My personal experience is that the lots that we've received from 3 different vendors that I've worked with in the United States are very consistent. And I'm not seeing this as a force for change in my quality system, but I am also doing the due diligence to do the checking. And if I did receive a lot of standards that was significantly statistically different than everything else, I would take action, which probably would just include sending that lot of standards back to the vendor and asking them to do an internal investigation.
Nice. So we have a question of a bud tender is trying to describe THC conversion to a patient as they read it on a lab result. Can you repeat your intro to that percentage? So this question I get asked almost on a daily basis, so I'll run with it. So the states have implemented that for potency, that you have to measure, the labs have to measure a certain number of cannabinoids, right? They also require that we determine what the total THC or active THC and total CBD or active CBD is in the product. And the reason why they ask for that is to give the consumer a better idea of the strength of the flower or concentrate that they're consuming once all the THC that's in the acidic form has converted into the active form. So depending on the sample type, for instance, for flower, most of the THC is going to be present in a non-active form. And that THCA weighs more than the THC. So when a consumer heats up their flower, they're converting that into the active THC, and thus they're losing weight along with that. So when we're giving a total THC, which is a percentage by weight, we want to account for that weight loss, that molecular weight loss during the heating of the product.
It looks like Jill reiterated that it was calcium and magnesium in high levels. So if it's calcium and magnesium, those are often in high levels, and they can also mean that your plant is a pretty happy plant because calcium and magnesium are used at, in their growth cycle, and they're not terribly toxic. I don't know what you mean when you say incredibly high. It would be, in my opinion, very difficult you'd have to take, I think, like a gram of magnesium, which would be a lot if you were selling a gram of weed and it had a gram of magnesium on it for you to get to a point where you might cause toxicity in a human. So compared to it not having any heavy metals present, that would be okay. Now, from a standpoint of, is that going to impact your concentrates? Is it going to have an issue with your flavor profile? Might it have an issue with a reaction that's going to happen further down the stream in your formulation? Having high amounts of non-cannabinoids present, like calcium and magnesium, could contribute to that. So from an internal quality standpoint, you might want to be concerned. But from a consumer standpoint, I don't think you're going to be able to get to the levels of calcium and magnesium that would be dangerous in such quantities on small amounts of flour. I mean, we were talking about having to consume pounds of flour all at once to get to that level. So hopefully I addressed that in a different way, since it's not cadmium and mercury.
Thanks, Tiffany. So the last question we have on here is that we mentioned front end and back end quality control methods as it relates to testing. Can you elaborate?
And so,
sure. Oh, you got this one, Garrett?
Sure, I'd be happy to. And Tiffany definitely brought it up in the beginning of the presentation as it relates to CIS-PQ and the formation of a quality management system within your cultivation or processing company. And that's all a factor of CGMP and GMP facility cultivation or processing when it comes to cannabis. Those are standards that the cannabis industry is aspiring to, but obviously because they're overseen by the FDA is not willing to give their full CGMP stamp of approval to any cannabis businesses. But when it comes to how to do, how to incorporate test results in those internal practices as it relates to the ways in which you cultivate and the way in which you process, it's important that from a standard operating procedure perspective, right, that you are taking these methods used in cultivation and processing from a method validation standpoint, wherein you're seeking to, as much as possible, define every single step of the process that goes into cultivating and processing these materials. And then you can use these test results to inform that process and to refine your own internal procedures in light of test results. So for example, proactively or reactively, right? Let's say we're doing a reactive scenario here where you get a failed test result What that should mean is that not just that the state's going to come in and seize that lot or take that batch and warrant in further investigations, but there should also be systems in place within your company such that when you receive a failed test result from your laboratory, that you initiate an internal review of the circumstances that led up to that failed test result, and that there are systems in place that are clearly defined for your business that determine who's involved in that investigation, what steps need to be taken to ensure that future batches don't fail for the same reason, and then following up in a feedback loop with the same laboratory to say, okay, we performed these, we made these process changes and integrated them into standard operating procedures. What did we fix it? You know, and the lab will help tell you that as well. For this reason, some companies are seeking to integrate testing capabilities into their own operations. The equipment's not cheap. So it's really large producers, either cultivators or processors, that are doing so. But that's all in support of internal quality assurance and quality control programs that they have incorporated. Because without one of those, you can't come anywhere close to calling your product GMP or CGMP.
All right.
I think I might just add to the front end discussion, because I think that was really excellent. Like back end, like what happens when you react, and there should be systems in place to do internal investigations. But from the front end, you have all these raw materials that are coming into your facility, whether it's your water, your soil, your
Absolutely.
nutrients, the packaging that you're using, all of the other raw materials that are coming in. All of those can be tested on the front end, saying, hey, guess what? There's no heavy metals in our water, and there's no heavy metals in our soil, and there's no heavy metals in any of the chemicals that we're putting into those as far as nutrients are concerned, maybe also pesticides or other chemicals that you're using. And there's no heavy metals in the container we're growing the plant in. And we're filtering the air in the facility, and we know that there's no heavy metals in the air that's in the facility. So our product is now on the front end, and we're monitoring all of these things through raw materials testing and environmental testing. Then we know from the front end that we don't have these issues. So from a quality control perspective, if you got them in your product, right, something has gone very, very farly wrong. You know, maybe an example for me is one time I was working in a pharmaceutical facility, and a ceiling tile fell into the product, and the ceiling tile hadn't been tested. Now this is, you know, an investigation had to be done. Hey, what happened to that ceiling tile? Where did it go? Why did it fall? Why didn't we not notice? Those kinds of things. But because we already had all of the raw materials, water, packaging components, utilities, testing in place on the front end, we could be sure that quality was built in before the plant even started growing, or in this case, before the tablet even started being pressed. So that's the front end side of the quality control method. It's a school of thought that says you build it in from the very beginning, and then your finished product testing is going to be consistent and the same every time.
Right.
Great. Well, I think that concludes all of our questions that we received. I'd like to again thank everybody who watched us today on our live webinar. So with that, have a great day, everybody.
Yeah, thank you all for joining us.
Thank you.